second part, we have talked a little bit about diversity, how we study that, and now we are moving to the limits of life. So life has colonized a wide variety of environments, including what we call stream environments. So we talk about the tree of life and how this life colonized different kinds of ecosystems characterized by different combinations of physicochemical parameters. I also mentioned before that archaea like its stream, so they are particularly well adapted to this kind of extreme environments, very acid, very hot, salty, or alkaline environments. So we will see some examples of that. And but let's start by the beginning and what we call extreme environments. What are they? And when we started to find life in these environments, because these environments at the beginning were thought to be sterile environments such as the Dead Sea. The name is very telling. And so in the 1930s, early 20th century, a guy called Eleazari Volcani started to see that there were microbes living in the Dead Sea, so it was not dead. And then the extremophiles became very famous from the 1960s and 70s. More or less, Carl Woese appeared in the scene at the same time, because we started to isolate microbes from hot systems. And of course, temperature is a major driver, determinant of communities, and a very important selective force. So very hot is very damaging. And so at that time, some people started to, actually Thomas Brock isolated Thermus aquaticus. RNA, the polymerase chain reaction is based on one enzyme initially isolated from this microbe. That's what we call it TAC. TAC polymerase comes from Thermus aquaticus. And then other microbes that grew at even higher optimal temperatures and up to 90 degrees. Sulfolobus acidocaldarius, at the beginning thought to be a bacterium, but then one of the initially described archaea. Okay, this is the start. So what's an extremophile? Starting by very basic things. So any organism on this planet or has grows at optimal conditions. So for any abiotic parameter, you have an optimum and then a maximum beyond which you cannot grow and you can even die, and the same for the minimum conditions for any given parameter. So you can pick a parameter, for instance temperature, and you can decide to establish boxes. Of course there is a continuum with the same principle, with an optimum, a maximum, and a minimum growth temperature or whatever parameter it is. And then you have limits upon beyond which organisms cannot divide or can even die. And so an extremophile would be an organism living close to the boundaries defining where life can thrive or not. So hyperthermophile, a psychrophile would be an extremophile, but not the others, or not that much, let's say. So basic idea, but just to be clear about that. So people have tried to classify the extremophiles depending on the different kinds of parameters that you can measure, and that there are many other parameters that you can look at, different types of chemicals. You can include them in this list, but in general, people try to think about major parameters that determine communities and are important drivers in particular in adaptations. This is the case of temperature, pH, salinity. You can include pressure, although it has not an important effect as temperature, for instance, but it has an effect. And then you have other series of parameters, desiccation, radiation, metals, where you cannot really talk about adaptation precisely to desiccation, to absence of water, but most likely about tolerance, and the same for radiation, the same for high concentrations of heavy metals, for instance. So there is a boundary also between adaptation to particular environments. You depend on these conditions to thrive, and tolerance to these parameters, with adaptation to low levels of water, for instance. So we will see some examples of this, some pictures. Okay. Why have been people interested in extremophiles? Very obvious, because of the molecular adaptations, the physiology of organisms, for instance, growing at very high temperature for biotechnological reasons, because many of the enzymes, the proteins that are produced by these organisms can function under conditions that may be interesting for industry, for instance, to treat waste or whatever other kinds of things. For basic science, for instance, the discovery of archaea was important, which are really adapted to to different kinds of stream, was important to understand partly the origin of life. Well, there's interest, I will mention that a bit later, but of course in evolution, for astrobiology, of course. Some ideas about extreme environments, that there is a decrease of diversity as you go to the boundary, really, of permissive conditions for life, that there is a predominance of microbial life. In these type of environments, micromineral interactions can be more apparent because there are less multicellular organisms, and so these kind of interactions may be more important. And many of these systems actually harbor many different extreme conditions at the same time. So many of these systems, and we will see examples as well. Okay. I will move a little bit around different examples to show you some of the features of these organisms, so more like a catalog, if you wish, but still useful. So hyperthermophiles, possibly the group of extremophilic organisms that have fascinated more scientists. So these are organisms defined to be to have an optimal temperature of growth higher than 80 degrees Celsius. And so here is one, it's one of the cases where you can see this idea that when you approach the limits of life, you have a reduction of diversity, and where you can see that archaea do better than others, because you have different types of archaea, bacteria, and eukaryotes thriving at lower temperatures. I mean, the eukaryotes can thrive up to 60, 65 degrees at most, some fungi, some different protozoa. Then you have archaea and bacteria growing up to, at the most, 95 degrees for bacteria, but beyond 95 degrees temperature and up, you only have archaea. So only archaea can thrive at temperatures of 100 degrees, say. Okay, environments like these ones, you have different kinds of hot springs. Some famous, this is Wolfram Zillig, so famous thermophilic guy. And then you have places like, and this one, deep sea vents and environments of the kind where you can have actually liquid water, which is important for life, at temperatures higher than 100 degrees. So it is in these kind of places where you can find organisms thriving up to temperatures around 115 degrees Celsius to maybe 120. I mean, the numbers, I don't know how reliable they are, but this is this particular strain of Methanopyrus candleri. This is a methanogenic archaeon, which is the record holder for high temperature. Well, it can thrive at 116 degrees at, let's say, kind of atmospheric pressure, and if you put some pressure, it grows even at higher temperature. Well, I don't know which is the reproducibility of that, but anyway, there are all the microbes that also grow up to this kind of temperature. So more or less, this is the upper limit for life in terms of temperature. And you find these organisms under pressure in the deep sea or the deep subsurface. The deep subsurface, there are different places, in the oceanic core, continental crust, and different, you can see that in the scheme. There are also hyperthermophilic viruses that affect, so wherever you have life, you have viruses and parasites, all the kinds of parasites as well. So these have been studied for some time, and they are peculiar. They have particular features, some funny shapes. So hyperthermophiles have been traditionally, and thermophiles in general, used for to generate or use enzymes for different things, purposes, in industry, industrial wastewaters, where you have sometimes high temperature, and you can work under those conditions to degrade different types of molecules, or do some bioleaching at high temperature. You have the famous PCR reaction that is based on the use of thermophilic and thermostable polymerases that work at high temperatures, but that cannot be denatured at 94 degrees or 95 degrees, when you temperatures that you use to separate the two strands of DNA to amplify them during this reaction. So this requires this kind of enzymes from thermophilic organisms or other kinds of molecules. And you have a list of enzymes. Some of them are commercial. You can buy them, so this works. This is just example, so we go fast. What about the molecular adaptation? The molecular adaptations have to do with how to cope with the deleterious effects of high temperatures. What do high temperatures do to molecules? Well, they increase membrane fluidity. That can be dangerous because if you increase too much membrane fluidity, you can have proton leakage, and any single organism uses a proton gradient across the membrane, or sometimes a sodium gradient across the membrane, to generate chemical energy through an ATPase. So if you have proton leakage across the membrane, you are not able to maintain a proton gradient, and then you cannot generate energy. So that's a big problem for cells. You have protein denaturation, of course, and you have thermodegradation and denaturation in the case of nucleic acids. So what are the adaptations faced to that? You have very different mechanisms. I mean, we don't need to go through all of them, but just to mention some. In the case of hyperthermophiles, you have, in terms of DNA, particular enzymes, in specifically reverse gyrase, that introduce positive supercoiling that compensates the denaturation, the opening of the double helix by temperature. You tend to denature, so the helix opens, and these enzymes will introduce positive supercoiling just to keep the same path of the helix for all the molecules to act under those topological constraints for which the different proteins in systems have evolved. You have protective DNA binding proteins and very efficient repair mechanisms. In the case of structural RNAs, RNase is, of course, with the double helix, it protects, and if you have a closed molecule, it cannot be denatured that easily. But the RNA, in general, in particular, structural RNAs are not closed molecules, and what they do is that they generally increase the GC content because you have 3 hydrogen bonds instead of 2, so that keeps the molecules tight and the structure stable. Usually you have more salt in the cytoplasm. Proteins in general tend to have more hydrophobic amino acids because that helps internal cohesion in molecules through hydrophobic forces. Or you have sulfur bonds between particular amino acids. And then you have a whole machinery that degrades proteins that are under degradation or don't fold well. This is chaperones and heat shock proteins. Okay, and in general, proteins are much more compact. So you avoid having loops that can be denatured easily. You have compact molecules with strong hydrophobic forces. And in terms of membranes, you have a whole range of adaptations in the phospholipids that prevent, or they rigidify membranes in general, and that prevent this ion leakage. And in that, archaeal lipids, which are very different from bacterial lipids in the nature of the lateral chain, which are isoprenoids, and also in the general in the glycerol phosphate isomer, which is 1-phosphate and not 3-phosphate. We heard about that yesterday from Connie, and the ether linkage. They confer this kind of rigidity to the membrane. So archaeal lipids are better adapted to high temperature. Okay, that's the thing. A word on the hypothermophilic nature of the ancestor, etc. This was an idea proposed very early on, based on the initial phylogenetic trees, in which the deepest branching lineages of archaea and bacteria appear to be hyperthermophilic. And so some people say, okay, this implies that the last universal common ancestor was a hyperthermophile, so growing optimally above 80 degrees, and maybe the origin of life was also at high temperature. This was highly discussed afterwards, because it turns out that with more diversity, we now know that these lineages of thermophilic bacteria, they are not at the base of the tree. They are well nested within the rest of the diversity, and we also know that they adapted secondarily to high temperature with the help of some horizontal gene transfer from archaea. But we do know that the ancestor, well, so the question is open. We do know that the ancestor of archaea was a hyperthermophile, and there were different adaptations to lower temperatures in different parts of the archaeal tree. The colors red means high temperature. By contrast, the ancestor of bacteria was likely not a hyperthermophilic organism. It may have been a thermophilic organism. So these are inferences based on on amino acid preferences. You can model that. This is some results. So this is a team of French people who have been doing this kind of exercise. They infer a hyperthermophilic ancestor for archaea, which can be also inferred from the topology of the tree of life, and the where the deepest branches correspond really to hyperthermophilic organisms. In the case of bacteria, the ancestor, they infer it's between 60 and 80 degrees. And so that actually may have been the temperature of the oceans at the time when these lineages evolved. Some people say so. At any rate, by this type of inference from genomes, you can also have you can also infer ancestral states for some proteins, and when you reconstruct these proteins in vitro and you test the activities, you get proteins that are active at higher temperatures, suggesting that maybe some of these organisms were more thermophilic than they should be. So there seems to be a contradiction between these two types of data. However, it has also been known for a long time that enzymes of hyperthermophilic organisms are much thermotolerant and thermoresistant than the organisms themselves. So these inferred temperatures for the specific optimal activity of different enzymes are not necessarily incompatible with inferences of lower optimal temperatures for the corresponding organisms. We are here at this moment, so maybe the ancestor lives somewhere between 60 and 80 degrees. Maybe life originated at those temperatures, but in particular if the oceans were at that time. But it's a hypothesis. So we have psychrophiles moving to the other extreme of the temperature gradient. And so organisms thriving at very low temperature. I mean, there has been some records of metabolic activity at very Low temperatures, minus 12, minus 17, some people have claiming even minus 20. But this is not temperatures where you can divide. So to me, this does not mean that life thrives at this temperature. That means that you slow down your metabolism and then you are still able to do some reactions, but that doesn't mean that organisms are living there in the sense reproducing. What I mean, when you are an organism has an optimum temperature or a maximum temperature at 115 degrees, it means that it can divide. It can develop the full life cycle at those temperatures. It's not the case at this temperature, even if you can have some metabolic activity. Different pictures of organisms, and you have some algae growing on snow. Well, so these things can thrive clearly in this type of ecosystems. And you have also enzymes working at very low temperatures, and that can be interesting also for a variety of things. So yeah, you have some examples here, not to read, but that exists. So how do these organisms adapt? Molecular adaptations, well, the effects of low temperature are the opposite of high temperature. So you induce higher membrane rigidity, and so you decrease ion permeability. So you can have problems also to create a gradient because you have this you need an optimal status of the membrane to be able to regulate your gradients. You can lead to protein inactivation, just simply proteins don't move anymore. So you need some kind of movement for enzymes to find the substrate and these kind of things. And then you can have, a limit in the terms of nucleic acid. The problem is that things are too winded, and that there is a limit that can represent a limit for an access limit for proteins that depend on processes like gene expression, for instance. If the helix is too close, proteins cannot get access to that. So what are the mechanisms in place? A little bit the opposite of what we see at high temperatures. You have mechanisms to unwind DNA because low temperature would tell to close the double helix. So you have proteins that introduce negative winding in the DNA that can be gyrase or different kinds of topoisomerases that act in cooperation with proteins such as histones. That happens in eukaryote, never mind. You can have anti-freezing molecules in the cytoplasm, things that prevent the formation of ice crystals. That is a big problem. If you have ice crystals, you can pierce the membrane and then you're gone. And that you can, in terms of proteins, what you do is all the contrary. You tend to increase the flexibility of molecules by having bigger loops and less internal intramolecular interactions. So more lateral chains, less charge, less hydrophobic amino acids in general. And in the case of the membrane, you have things that increase membrane fluidity, and here bacterial lipids are more adapted than archaeal lipids. So, as a summary, what we have is the opposite effect. In terms of DNA, RNA, you have positive supercoiling or negative supercoiling. High and low temperatures, you have more compact proteins, more interactions, intramolecular interactions, both ionic and hydrophobic forces. Here on the other side, you have looser proteins and less intramolecular interactions, and lower permeability, higher permeability membranes. Okay, so that's the general idea, very basic. Halophytes, so these are organisms adapted to high salt concentrations. You have some examples here, and including salt saturation, which in the case of sodium chloride arrives around 30%, a little bit more. And so these beautiful red pinkish colors are due to halophilic archaea that sometimes can form the nucleation center for halite crystals, like here, and these can stay for a long time, and then you can pick for years even, and then you can pick them and let them grow again. So among the most halophilic organisms, you have these funny species, or Haloquadratum species. This is the name of a genus, Haloquadratum. You can tell from the shape why it is called so. And this is a funny archaeon that is very flat. Actually, you can see here by fluorescence, this is a cut of the same cell. And all these little dots here are vacuoles, gas vacuoles that help to orient the cell, that actually acts like a solar panel, because these organisms, they are not photosynthetic, but they are phototrophic. They have a particular protein that gets them some energy from light. So they can get ATP from light, but they cannot fix carbon. And so they really act as solar panels, and that's hence the shape. It's kind of convergence. It works quite well for them. So how do these organisms adapt? Well, there are different ways. The most halophilic organisms, in particular archaea, accumulate potassium up to 4 molar in the cytoplasm. Other organisms that are less halophilic, in particular bacteria, but also some archaea, have that as an additional source, have compatible solutes. These are small amino acids or things like betaine, small sugars, that increase the viscosity of the cytoplasm. So in a sense, you need to cope with the osmotic forces across the membrane. And so you have also transporters that pump out sodium, and in some cases pump in potassium, in the case of halophilic archaea. And you have proteins that are in general charged negatively, just to keep the structure in such an important amount of cations. Sodium in the case of the extracellular surfaces, or potassium where you are in the cytoplasm. And also the cell surface is charged negatively, in particular with lots of exopolysaccharides. And one typical thing is that you have an acidification of proteins in general. That's what I say here. The proteins are have an excess of negatively charged amino acids, and so these are acidic amino acids like glutamic and aspartic acids, and this can be shown by this kind of curves or of the pi of proteins, so isoelectric point of proteins, and the percentage here. And you see that this is for different proteins inferred from metagenomes. So this is the collection of all proteins for different types of organisms. And normally, organisms like you and me, we have a bimodal distribution. But as you get more and more salt, you get a unimodal distribution with a shift towards low pi values, which means that these proteins are highly acidic. I will give you another example later. So alkaliphilic organisms, we now move to pH as an abiotic parameter that can acquire limiting factor levels for life, although, well, we know organisms that adapt well. So you have here pictures of different systems that are alkaline, so some lakes. Some of them are also hypersaline. Here, and you have lakes like Calchichica. Someone is studying this in the system in Mexico. This is an alkaline crater lake with beautiful living microbialites, and Gliomargarita litophora was isolated from this lake and precipitates carbonates inside. So different types of systems where you have different molecules. On the other side of the pH gradient, you have acidophilic organisms that adapt or thrive at very low pH conditions. You can set it at 2, 2.5 or less than that. These are images of places where we find such conditions. This is Rio Tinto in Spain. This is also Rio Tinto, but this is Rio Tinto, but this is in Japan, a place where the organism that is known to be the most acidophilic was isolated from volcanic sources. This is Picrophilus oshimae, living an optimal pH of 0.7, but able to thrive at slightly negative pH values. An archaeon for sure. Many of these organisms, but you have very many bacteria adapted to thrive in these particular systems. This is the case of Acidibacillus ferrooxidans or T-oxidans. There are several species that are particularly adapted to thrive at pH values such as 1 and the like, and they can use, they can mobilize metals. So these type of organisms have been used traditionally for bioleaching, for the extraction of metals from minerals, because they gain energy. Actually, they are quite versatile. They can gain energy from the oxidation of iron, the reduction of iron, and other kinds of metals. So they have been traditionally used for mining or helping mining in different systems, just by watering piles of mineral with natural cultures of these kind of organisms. And we have now genomes and can infer the molecular adaptations of these organisms, which in general, what happens is that they can function at very low pH, but the internal pH of the cell is close to neutrality. So they have mechanisms to keep intracellular pH much higher. Even Picrophilus oshimae, which grows at pH 0, basically, has an internal pH value in the cytoplasm of 4.5. It's slightly acidic, but it's much higher of the pH in the surrounding medium. And so, yeah, they are very well known, in particular in acid drainage systems. Okay, some pictures about birophiles or piezophiles, perhaps a more appropriate term, organisms that live better at high temperature. Some of them can also live at at atmospheric pressure, even if they do less well than at higher pressure. There are some people studying this, and even in the audience. And where do you find these kind of organisms? Of course, in the deep sea, but also in the continental and oceanic crusts. And then, well, they do things in the ecosystem and help to keep the biogeochemical cycling in these deep oceanic areas. Okay, xerophiles organisms. Here we define this boundary. We need water, so organisms still need water under those conditions, but they are adapted to very low water conditions. And so these are organisms found typically in deserts, living with really, very, very low hydration states. Some of these organisms, in particular, adapted that are exposed in these areas to ultraviolet light coming from the sun, can be also highly radiotolerant. And this is the case of Deinococcus radiodurans, that is not an organism adapted to radioactivity, but it turns out that it works, that it can resist well, just because it's a xerophilic organism. It is usually in environments where you have a lot of irradiation, and so the same adaptations that are needed to cope with UV light work very well to resist other types of radiation, including having many copies of the genome and strong repair mechanisms for DNA, which is one of the major damages of UV light and also other kinds of radiation. In these particular systems, you have many organisms that adapt by finding particular conditions where you can have the condensation of water or reach a higher hydric state. So in this kind of desert systems, cold or dry or hot, you have cryptoindolytic organisms that live within rocks. First of all, that helps you to be protected from UV radiation, but also you can have the condensation of water molecules in particular parts of rocks where to be able to thrive for some time. And in particular, this has been studied in the Atacama Desert, where researchers have shown that in some areas, in particular in some allied places of the driest part of the Atacama Desert, you can have occasionally during the year condensation of water within rocks, within allied, but not outside. And these, during these hydration events, organisms, in particular cyanobacteria that are halophilic and thrive there, can grow for a little bit and then enter dormancy again. So these are local episodes of water condensation that, you are really at the boundaries of what is permissive for life. When there is no water, you just sleep, and when there is some water, you can maybe reproduce once. You have also the deep subsurface. I should have put this closer to the pressure, maybe. So an environment where you find different types of extreme conditions, including pressure, including sometimes also high temperature, because the deepest you go, the higher the temperature, and in some places you can get really high temperature. So maybe in this type of environment, the limiting factor is temperature. Pressure, I'm not that sure, but another important limiting factor is the sources of energy that you can get. So one environmental type of condition that can be really limiting for life, it's what we call oligotrophic conditions, where you have limiting access to nutrients, and that can be a problem. But still, you can have organisms thriving there, and organisms in the deep subsurface, I mean, it's difficult to study, you have problems of contamination, etc., but projects or initiatives such as the International Ocean Drilling Program have been working over the years to try to identify organisms that still live in the deep ocean, for instance in sediment, below the surface of the sediments, by drilling and then studying things, different layers, of course. And you can show that there is some activity of archaea in particular as you go deeper, and more in particular molecules associated with this archaea. Okay, well, so this raises things as or questions as is there a subsurface ecosystem on Earth that is based on the abiotic hydrogen production and is disconnected from the surface? This is a question that regularly comes on the table. So maybe not. So this is a different question, but because finally different oxidants that these organisms can get there may come from the surface. So there's a question, but it's an open question so far, so maybe. And that would somehow make the question of, is there life in the subsurface of places like Mars possible if we have similar conditions? Open question, I don't know, but you can reasonably ask it. Okay, so I've talked about adaptation to a variety of extremes. We can also talk about the resistance form. This is different. So we have, of course, yesterday we heard in the documentary that there were tardigrades. I don't know if they can resist to 150, but 110 maybe yes, because they are not in nature, but they don't thrive under those conditions. So you need clearly to separate one thing. There is adaptation to particular conditions. You have your own, your whole life cycle there, and then resistance. So you can simply produce spores and stay in dormancy for a long time, and then you can resuscitate the cell if the conditions are again optimal or permissive for life. And you can also get preservation over long term by natural conditions without the need to make spores on any particular kind of structure. There are two major methods to preserve things over time. One is cryopreservation. I mean, even humans, you can freeze spermatozoids and keep them for a while and then use them for fertilizing eggs or eggs. Ovocytes, for instance. Nature does that very well. You go to permafrost today and you can resuscitate organisms that were living thousands of years ago and make them grow again. So it works very well even in nature. And you have lyophilization, so desiccation. That's what we saw in the halite in the halite rocks in the Atacama Desert. When the conditions, when water is not there anymore, you get desiccated. Many organisms are very well adapted to do so. And then if you hydrate again your sample, you will have them growing again. So that's that has to do with dormancy. Resilience is the resistance over time to that. And that helps organisms to cope with things like exposure to a different variety of damaging surface conditions, including spatial radiation in some kind of exposed experiments in the International Station. So some of these organisms can resist for a while this kind of radiation when they are in dormancy in these kind of systems. Okay, and these, of course, these kinds of the conditions where you find organisms define the limits, at least known on Earth, within which life can thrive. And if life exists in other places in the universe, it is more likely based on similar organic carbon with liquid water. So we can expect that more or less the same type of factors can be limiting for life. You can vary a little bit the boundaries, but in principle, we expect more or less the same boundaries for any form of life out there for some form of life that is based, of course, on organic carbon and liquid water in terms of salinity, temperature, etc. So by studying the limits of life on Earth, you can push or extend the limits of life or the type of, you can define the type of systems where you can reasonably expect to find life elsewhere if it ever evolved there. And so I have been talking a little bit in general, and then there is the question of poly-extreme environments. What are the limits? Because as I mentioned at the beginning, in many environments you have combinations of different conditions. So here I'm going to talk about some work that we did in very particular environments on Earth to define the limits to face to different parameters such as high temperature, high salinity at very low pH. So this is an example. So as I mentioned before, salinity, high salt, it depends on the type of salt. So saturating conditions for NaCl, sodium chloride, is around 5 molar or 35% weight volume, sodium chloride. And then a wide variety of organisms, in particular archaea, adapted to these kind of salt conditions, and this includes different types of family, genera, whatever classes of archaea. But different salts have different solubility. For instance, magnesium chloride saturates at 66%. And this means that bitter brines that have a lot of magnesium chloride have low water activity, much lower than NaCl. And in many cases, these salts are highly chaotropic. This is the case for magnesium. What is a chaotropic salt? These are salts that weaken intermolecular or intramolecular and intramolecular interactions, van der Waals forces, etc. And so this tends to disorganize macromolecules. So there's many anions and cations that are known as chaotropic at different levels. Magnesium and calcium and iron are among the most chaotropic. So they have been studies about which are the limits to chaotropicity and water activity in the lab with cultures, but there are many microbes out there that are not in culture. Can life cope there under conditions beyond that? Of course, to study that, you need to have environments where these conditions coexist. And this is when I start talking about this place, the Dalol protovolcano in the Danakil Depression. This is in Ethiopia. It is located in the Afar Triangle, so this is a very active place in terms of tectonics. We are at the confluence of 3 tectonic plates, so there is rifting, and with that we have associated widespread volcanic activity. So all along the East African Rift, so this delimits 2 tectonic plates, we have volcanoes, and this is the case here. Along this Danakil Depression, you have the Ertale volcanic chain, and there are volcanoes up here. So a lot of volcanoes and a lot of hydrothermal activity associated to these volcanoes. This is one thing, but at the same time, it is one of the hottest areas on the planet. It is 35 degrees average, annual average, so it means that in winter during the day you have 45, 50 degrees without problem. And this means that you have a high evaporation rate. And so this led over time, after the closure with the emergence of these 2 volcanoes that closed the entrance of the Red Sea in the area, this led to the evaporation of seawater. There were several episodes, and this led to the accumulation of a thick salt crust that is 1 to 2 kilometers, and that lies at the bottom of the Dalol Depression. We are below the sea level here, around the deepest point is minus 120 meters. So we went to this system and sampled it. So how it works, so Dalol is not a volcano, it is a protovolcano. It will be a volcano in the future, possibly, but there is not yet lava emission. What you have is a mixture of hydrothermal fluids that are very salty. Why? Because so you get the water that comes from the high plateaus in the Ethiopian part. Mostly you have very high mountains. This water interacts with a magma chamber that is quite close to the surface, maybe 1 to 2 kilometers below the surface below the Dalol Dome. And there, these fluids traverse the sediments from the marine sediments, the old marine sediments, and then the thick salt crust, and they get enriched in different chemicals, giving rise to different kinds of hydrothermal reservoirs on the surface that have slightly different chemistries. So you have molten salts and also highly salty fluids, let's say. So this is an aspect of the real picture of the satellite picture of the Danakil Depression. Dalol is there, and this means that you have here conditions that are very diverse, from the Lake Asale or Lake Karum at the south of the Danakil Depression to different reservoirs, hydrothermal reservoirs at the Dalol Dome and around the Dalol Dome. In the salt plain, and different hydrothermal manifestations, the Yellow Lake, the Black Lake. We will see pictures now, the Western Canyon lakes that are active hydrothermal systems and then have different chemistries. So some pictures, this is how the Dalol hyperacidic and hypersaline hot ponds look like on top of the dome. It's a very active region. So we have pH that is slightly negative, so minus 0.2 to minus 1.5. The colors are not due to biology, this is chemistry. You have lots of iron. The fluids get out of the chimneys at a temperature of around 108 degrees. So these are salt chimneys, they are very salty, so enriched in many chaotropic salts, magnesium, calcium, and iron. And it is iron which gives this color. So the fluid is transparent as it gets out of the chimney, but as iron gets oxidized, it gets to these orange colors, and so passing through the greenish, etc. There are many other elements there. So the Black Lake, this is a very peculiar place. We have a temperature of 70 degrees, more than 70% and up to 90% salt saturation. It's essentially magnesium chloride, so extremely chaotropic environment, pH 3. We have the Yellow Lake. It's actively bubbling, so you have lots of, there are also many organic compounds and gases like ethane, butane, short hydrocarbons. So if you work for some time, you really need to wear masks, otherwise you feel sick. So you have also very high salinity, over 60%, extremely chaotropic salts, lots of calcium in particular in this place. Temperature 40-45 degrees and pH almost 2. There are some places that look milder. You have the salt plain that can be inundated depending on the rain regime during the year. So it can be affected by hydrothermalism. You can tell that from the yellowish color here. pH 4, temperature varying depending on the time of the day, 30-50 degrees more, sometimes salinity, saturation, of course. Then you have some underground reservoirs in some caves. There appear less extreme, pH closer to neutrality, bad salt saturation. And we have the Western Canyon lakes looking like the Yellow Lake, so you have a lot of bubbling and degassing, but somehow milder conditions, less acidic pH, and slightly less saturation, although also extremely salty. So this all represents the gradients of poly-extreme conditions, whether which to study which are the limits of life, moving from pH minus 1.5 more or less to circumneutrality, temperature between 30, 110, let's say, salinity, always saturation, but sometimes really super saturated, and very different types of salts, chaotropic salts included. So what are the physicochemical limits of life? And second question, what are the molecular adaptations to this particular system if organisms are there? So we did a wide variety of things, collected many samples from different places, and we analyzed the chemistry of brines. We tried to culture microbes, did metabarcoding analysis, metagenomic analysis, cell sorting, and different types of microscopy, so a battery of analysis, always using negative controls. It is essential because contamination is a problem. So to make a long story short, we have plentiful evidence of life, microbial life in these systems. We do not have unambiguous evidence for life in these other conditions. Using the appropriate controls for that, that is very important, because if you go there, you can get all kinds of contaminants, including from... So with that, what we can conclude? We can conclude that we have systems on the surface of the planet Earth, an inhabited planet, that have liquid water, but that have no life. So that might have implications for habitability, even on Earth, but somehow elsewhere too. That's one thing. And then how the organisms that live in the most extreme conditions adapt, and what kind of organisms are they? Well, they are dominated by archaea in the most extremophilic polyextreme environments, the Western Canyon lakes. More than 99% of the organisms there are archaea. And in particular, they fall in two different groups. The halobacteria, this is an old name, it's haloarchaea, belonging to the group Halobacteriota. This dates back to the time when these organisms were thought to be bacteria. And then the nanohaloarchaeota, And there are some different kinds of bacteria. So 2 major groups, but they are they're very, very diverse. So, and this is shown in this phylogenetic tree where all the branches in blue come from our sequences, and the red dots as well. These are Sanger sequences. This is metabarcoding analysis. We can also tell that some of these organisms are hyperthermophilic because, as I mentioned before, one of the adaptations of structural RNAs to high temperature is to increase the GC content just because you have 3 hydrogen bonds. And actually, you can have a very good correlation between culture species adapted to different temperatures and the GC content. And in orange, you have the different sequences obtained from our systems, and many of these sequences fall in the hyperthermophilic or thermophilic part of the spectrum. So these organisms are likely hyperthermophilic, and then others are thermophilic. Now, I mentioned something that, I mean, there are 2 major types of organisms here. There are halophilic archaea and there are nanohaloarchaea. So these are organisms that are parasites or symbionts of haloarchaea. Now, in principle, they lack from their genomes or metabolic pathways to synthesize amino acids, phospholipids, and other things, so they need to pump that from their hosts. They are inferred to be possibly parasites, but some of these organisms have been kept in culture, like this Nanohalobium costans, and in some particular cases, some of this parasitism can evolve into mutualism. I mentioned that before. And in particular, some of these parasites have some genes that allow the host to exploit a particular organic substrate. In particular, in this species is lignin. And so, in the absence of any other carbon source, the host needs the parasite to exploit a particular substrate. So this is a parasitism evolving into a mutualism depending on the environmental conditions. So the environment can shift the relationships, the biotic relationship among organisms. So that's an interesting case. So I've talked about life, and I would like to mention 2 things that are important, especially because this applies also to finding traces of life in the past at the time when life evolved, but also if you look for life in other planets or traces of life. You have to be careful about 2 things, and which are problems to try to detect life or the absence of life at the boundaries of life. One of them is contaminants. So we know that there are we can get false positive in very low biomass environments because there are plenty of bacteria in the air, in our hands, so we can contaminate our samples. That can be contaminated during laboratory work, even if you work properly and they're in sterile conditions. It's still a problem. You can also have microbial contaminants in kits that you buy to companies that is called a chytome. And you can also, of course, have contaminants that are natural, coming from instance from dust particles that are moved with the wind. And that happens typically in Dalol. And so to try to estimate the kind of contamination coming from natural sources in the area, what we did is to place filters for a couple of weeks. They are on place, and after these 2 couple of weeks, we took these filters and we analyzed the diversity of things that had fallen on the filters. And then we analyzed that by metabarcoding. So, and what you see in this slide is the diversity that you find in those filters, and that from organisms that are thriving in places where sure you have life in that kind of systems, and they are dominated by archaea in red colors. You find some of these archaea in the contaminants coming from with the wind, because you have salt around, and then you have organisms even coming from the sea. So within this organism, you have some typical marine bacteria, but also lots of organisms typical of soil, because they come from the dust around in the mountains, and you have very strong winds in the area. So you can detect this contamination. So the question here is, why? If you have all these organisms coming all the time, why don't you detect them in the Dalol ponds when you work properly? Okay? And the answer is very simple, and it is that these Dalol hyperacidic brines degrade DNA and cells, and you can show that experimentally by incubating different cultures with a particular brine coming from the top of the dome, cultures of E. coli, but also of Halobacterium salinarum. So this is a halophilic archaeon, or just communities, PS, communities coming from the salt plain that are living very nearby. And so when you incubate or you put in contact the brine with this organism, the DNA, the concentration that you DNA from cultures decreases immediately. So this means that cells are being lysed. You can no longer amplify your RNA genes. You don't see anything after the brines have been in contact with cells. And you can also see that under the microscope, epifluorescent microscope. So these are natural communities from the cave in the Dalol system and from the Lake Casale. These are colored in cyber green, so this colors the DNA, and so you see different dots, not many because there is a light, but there are dots there. And when you touch that with the brines, all the signal disappears, so it means that the cells and the DNA are degraded. Okay, another big challenge of detecting life at the limit of life, I will mention the problem of biomorphs. What are biomorphs? Biomorphs are things like that look structures that look like life but are abiotic, are mineral. So in this system we have cells, you can see them, these are different real cells, these are not cells, these are crystals, halite crystal, but then these are cells, clear cells that have some cytoplasmatic bridges, so different kinds of cells. But you have also biomorphs. So on the left here you have a package of cells, they are small, and these are silica biomorphs. They are the most typical. You have also some sulfur, and they look like cells. The size and the aspect is the same, but these are mineral particles. So it's very easy to have a look under the microscope, and if you're not aware of that, you can say that these are microbes when they are not. Furthermore, these crystals or these precipitates can chelate, can absorb molecules, including nucleic acids and probes, and this can be seen, I mean, the light is not appropriate for that, but this can be seen here in this picture where these are mineral structures coming from this Dalol system where you can use stains typical of that absorb the dye DNA, and that are here just showing mineral structures, and it is the same for oligonucleotide probes. And you have many different types of shapes for these different kinds of biomorphs. They are mostly made out of silica, but you have sometimes magnesium chloride, and so these are all confounding factors that can induce to error if you are not critical enough. Okay, now how do these organisms adapt? Now moving on to more fine adaptations. So in order to analyze that, we sequence metagenome, so we have access here to the protein coding genes, and we generated MAGs. Now you know what are MAGs. So these are metagenome assembled genomes. For most of them Haloarchaea, but there are also some bacteria in these different systems. And I just recall the adaptations of halophiles. There are two major strategies. Moderate halophiles tend to use a salt out strategy. They just eject sodium out of the cell, and they try to produce some compatible solutes to increase the cytoplasm viscosity. Extreme halophiles, they use a salt-in strategy. They pump in potassium up to 4 molar. As I mentioned before, this goes with an accumulation of negatively charged amino acids, acidic amino acids such as glutamic and aspartic, and a depletion also of hydrophobic amino acids. I didn't mention that, but I mention it now. So what do we see in these systems? Okay, this is just what I showed before. These are PI values inferred for different proteomes. I mean, so we have metagenomes from freshwater marine systems, and different solar salters are increasing salt concentrations. So you see that you shift from a bimodal distribution. So these are all the proteins from all the genomes there. So it's a lot of proteins, thousands of proteins. And you see this bimodal distribution, and when you have 32% salt, there is a peak, a unimodal distribution. We have seen that before. Now, what we have in Dalol, we have this unimodal distribution, but much higher in intensity. In some cases, in particular in the Western Canyon lakes, we have more than 80% of proteins that are extremely acidic with very low PI values, okay? And this is the same thing in another form, okay? And so you can see that at the level of amino acids. So if this is a principal component analysis, and you have here the different amino acids that are called with single letters, and you have here different, in particular, pay attention to this DE, which is the acidic amino acids, aspartic and glutamic, IK ratio, so versus some hydrophobic amino acids, and you see that these Dalol systems correlate very well, and the sea water and all the more mesophilic ecosystems are very far away. You can see that also at the level of individual genomes. So these are max of archaea and bacteria, and here you can tell that archaea do much better than bacteria. So the older proteome is very acidic as compared to the bacteria that also can thrive in this environment. So they are already quite well adapted, but not as well as archaea. And you can see the same trend when you go from the less polyextreme environment, Lake Casale, Lake Karum, or towards the Western Canyon Lake, where you have really very push adaptations, the most acidic proteins that you can get for a particular microbe. So we can also analyze the functions of these archaea. I'm not necessarily going into the details, but what you can see from the prediction of the genome is that they are very flexible. They can use the diverse, they are heterotrophic, they can use diverse sources of energy and carbon in the system, and they can be aerobic or anaerobic depending on where they are. So they can shift between different energy gaining modes depending on the function. One of the things that they have is that they can degrade alkanes and haloalkanes, and also a wide variety of carbohydrates. These are organic molecules coming from the sediments that are mobilized with the fluids. And one of the things that they can do is that they can produce and degrade polyhydroxybutyrate. This is a carbon storage form, and this is interesting because this helps these archaea mostly to adapt to a very peculiar lifestyle that is very very useful in this kind of settings. And this is a feast and famine strategy. The feast comes when you have the hydrothermal fluids bringing the organic molecules, and they have nutrients. They provide electron donors and acceptors, and so these archaea can thrive there. There is also hydration. And the famine comes when you have desiccation. So these organisms get trapped progressively within the salt crystals until full evaporation of the system, and they need to thrive from their own reserves, carbon reserves, until the next feast. Okay, so this is particularly well adapted. And so I'm close to the end, I guess, but I would like to mention that I was not sure whether I would tell this story or not, but among the diversity that we have found in this system, we have detected new lineages of hyperallophilic archaea. And this, I'm not going to enter in the details, but this, the discovery of these lineages is interesting because these lineages are branching deep at the base of the archaea, or they are branching deep as a sister group to the classical nano-haloarchaea. And so these lineages that branch deeply in the tree of life can help us to solve evolutionary questions. And one of the questions that has been asked for a long time, in particular in the case of archaea, is whether there was just one adaptation or very few adaptations to high salt, or there were many adaptations, independent adaptations to high salinity. So this is a phylogenetic backbone, and some studies place nano-haloarchaea at the base of classical haloarchaea or in a different place of the tree. So which is the right solution? Depending on where you place this nano-haloarchaea, you can have one single adaptation or multiple adaptations to salinity. So this is more or less the question. There are different ways in which you can improve phylogenies. One is to have more data, and both in terms of sequences, but also in terms of taxon sampling. So we talk about taxon sampling here. So having more groups can help to better solve the backbone tree of archaea in this particular case. And the other thing that you can do is to use better models of sequence evolution that can cope with differences in evolutionary rate, that can help to avoid phenomena such as the long branch attraction, which is one big problem in molecular phylogenetics, and compositional biases. And of course, you know that these archaea have very strong compositional biases because they have lots of very acidic amino acids. So that is a problem. And you can see that this affects differently different kinds of proteins. So depending on the proteins that you use, you can get different results. So this is just to show you this different biases, in particular in different data sets that people have been using to reconstruct the phylogeny of this archaea. So this is a data set based on ribosomal proteins, 48 ribosomal proteins, and this data set of proteins for different groups of archaea, as you can see, have very different composition in terms of amino acids, so the different vectors here, from the rest of the proteins of the proteome. Now, there are other data sets that, in particular, we have used in the past that better represent the whole proteome in the system. So these kind of proteins are better adapted than this to solve this question. So if you mix everything together, and you include these new groups, this more taxon sampling in the system, you can get a better result phylogenetic tree for these kind of organisms, and then you can infer that at least that the adaptation to extreme allophily in archaea happened at least 4 times in this particular tree, at the base of the classical haloarchaea, at the base of the methanonatronarchaea, we haven't talked about that, and haloplasmatales, they are not very frequent in ecosystem, and at the base of the nano-haloarchaeota. And then this, there was a phenomenon of convergence adaptation, so selecting, for instance, for more acidic proteins with horizontal gene transfer, sometimes from classical haloarchaea to the other archaea. And horizontal gene transfer works very well in evolution. And there is possibly even more independent adaptations. We also detected a new family of archaea, in particular belonging to the DPAN supergroup, for people who know about that, that also has this unimodal distribution and seems to have adapted secondarily and independently to hyperallophily. And this will be, I think, my last slide, or almost so. It's not only archaea, there are also viruses in the system, and it is even more complex than that because there are even viruses of viruses. So these are called virus satellites. So you can infer from these metagenomes and there are new lineages of viruses in this system belonging to different kinds of families. And so you can infer that there are viruses infecting Haloarchaea, infecting nanohaloarchaea, so the parasites of Haloarchaea. So you have nested systems of symbiosis and parasitism in these kind of environments, and you have satellite viruses using other viruses to move around. So that's the complexity of biology in all kinds of systems. And that's the end. So this is just to thank people of my team or our team, some of which are here over the time, people who have been sampling with us in the field in different kinds of extreme conditions. And thank you for your attention.